So what we're talking about today, largely, is about drugs which work on the serotonin 5-HT2A receptor. And there's a remarkable relationship, the affinity of the drugs we're talking about. We've talked about LSD and psilocybin so far. There's mescaline over there from the peyote cactus. They all bind to the 2A receptor, and the affinity of them for that receptor is strongly predictive of clinical dosing in a way which is really quite remarkable. It actually tells us, just simply from that correlation coefficient, that these drugs all work through stimulating the receptor, or the binding to the receptor, and they act as agonists, they stimulate the receptor. And if you go to the fireside chat with Brian Roth later on today, he'll tell you how complicated and sophisticated that process is. So I'm not going to touch a bit on it at all, because he's the world expert. And of course, you've already heard from the presentations from the beginning of the day about mystical experiences having a profound predictive value, at least in that particular study, in terms of outcome. That's the mind end of things. So I thought it might be quite useful to talk a bit about the brain in between. Okay. So, aha, great. Okay. So the 5-HT2A receptor is a really interesting receptor because it's particularly expressed in brain, and it's particularly expressed in the parts of the human brain which are most recently evolved, i.e. the parts of the human brain which make you human. And you can see this is a heat map, and the hotter it is, the more receptors. And I can tell you the green bit at the top that's the sensory motor cortex, and that explains why with relatively few receptors you get relatively little impact on your ability to, particularly to move when you take psychedelics. But those red bits at the front and in the middle there, those are the parts of the brain where you do your very high-level thinking, planning, reflecting, imagining, etc. So the human elements, or the key elements of human experience, and very few receptors lower down in the brain, like the midbrain, the thalamus, etc. And the human brain has more of these receptors in terms of density per unit of tissue than any other species. And some people believe that they've actually driven the evolution of the large size of the human brain. That's hard to prove. Probably not true, but there's no question they have a very important location in human brain function. And they are expressed on neurons, and they're expressed particularly on a collection of neurons which are called the layer 5 pyramidal cells, and they sit in the cortex. These are images from probably rat cortex, showing the distribution of neurons and the high density of these receptors in this layer 5. And the layer 5 pyramidal cells are powerfully depolarized. You can see on the right-hand image there, this massive depolarization of these cells when a 5-HT2A agonist is given. And those cells are the neurons which regulate cross-cortical functioning. As you know, you've got probably 100 billion neurons in your cortex. They have to talk to each other. And the talking between the different parts of the brain is coordinated by these layer 5 pyramidal cells. And if they work differently, then you think differently, because they coordinate the very high-level processing that is involved in things like thinking. And the concept of top-down control is one that we will talk a bit about, I'm sure, throughout the day. The idea that the brain creates plans and images and projections and inferences, and then tests those theories in relation to sensory inputs and motor outputs. And that top-down control is fundamentally dependent on the layer 5 pyramidal cells working in the efficient way that they have tended to learn to do over the decades in which your brain has been working out how to work. Now, there are a number of impacts on brain function that psychedelics produce. So there's a little review there you can read, again showing the density of the receptors in the brain. And there are three things happen when you, as I've already mentioned, you get a psychedelic, it stimulates the 2A receptors, profoundly depolarizes those layer 5 pyramidal cells. That then results in an extraordinary perturbation of ongoing oscillatory activity in the brain. And you can see there in the middle box, you can see the normal brain rhythms, maybe the 10 hertz alpha rhythm, completely fragmented instantly. A psychedelic gets into the brain. And as a result of that, the breaking down of ongoing regulated function The brain becomes more connected, and that's the two circles higher up show the increased connectivity of the brain, which is induced by the breaking down of the ongoing activity that's normally present. And then on the right-hand side, there's this fascinating new insight, which is the ability of the 2A agonists to stimulate neuroplasticity, by which we mean they can increase the branching and the ramifications of dendrites, those curly things that come out of the top of the pyramidal cells, and also increase the number of synapses, the little blobs that are seen on that sort of tadpole-like image there. And those synapses, of course, are critical for making new learnings, new memories. And one of the reasons why psychedelics probably produce such enduring effects is because they facilitate the laying down of new insights, which is likely to underpin the reason they're so therapeutically useful. So here's a series of experiments that we've done in recent years looking at the brain signatures of a range of different drugs. And this is using a technique called MEG, magnetoencephalography. And you can see that different drugs have profoundly different effects. And I've put in the red box the 3 psychedelics that we're likely to be talking about most today. Well, we're certainly talking about psilocybin and LSD, but we'll also be talking later on about ketamine. And those drugs produce that blue coloring. And so blue means a reduction in the power of the oscillatory activity, whereas red means an increase in the power of oscillatory activity. So on the left-hand side, you see GABA drugs like zolpidem, the sleeping pill, gaboxidol, which is a GABA agonist, tiagabine, a GABA uptake blocker, propofol, an anesthetic. They all produce profound increases in synchronicity in the brain. The brain becomes locked into rhythms, which, in the case of propofol, is what anesthesia is. It's where you're not conscious because your brain doesn't have enough flexibility to do consciousness-type things. But on the right-hand side, you see the opposite. You see the blue coloring tells us there's a massive reduction in power in those oscillatory circuits. And that is that's a term we often use to explain that, is entropy. The brain becomes much more chaotic, much more entropic. And that probably is the critical factor in changing the way people think as a result of these drugs. And one example of that is here. So this is our MEG study, the first MEG study we did with psilocybin, looking at the power of the alpha rhythm in the posterior cingulate cortex, and this very powerful relationship between the disruption of that oscillatory power and the experience of ego dissolution. Really very compelling evidence that this may be, the change in oscillatory function may be driving the subjective experience. So let's try to make sense of it from what you think about and understand how the brain works. So I'm going to use the visual system as an example because it's the one that's very well studied, and we can say with some certainty quite how the brain creates images. Your brain isn't a camera, by the way. For those of you who think your brain is like your phone, it's not. Your brain is not a camera. Everything you're seeing, i.e. me now, is a construction, an estimate that your brain makes of what's likely to be there. So the way it does it is that photons from the outside world go into your eye, they stimulate neurons in the retina, and then a whole series of electrical impulses go up the optic nerve into this large chunk of the brain, which is responsible for the creation of a sense of vision. And the brain reconstructs an image of what it expects to see, and you expect to see me, and in fact, if I was to change dramatically whilst I was talking, most of you wouldn't even notice because you know that's impossible. So you'd carry on assuming it was me even if it wasn't, because that's how the brain works. It makes an inference about the world, and then and if it tests that, and if it's right, it doesn't bother to re-upgrade it. And that's why the brain is 10 times more efficient as a computing machine than any current computer. But after drugs like psilocybin or LSD, it cannot reconstruct the images in the same way. And for periods during the trip, the disruption of those layer 5 neurons means that you cannot turn the external input into the typical image that you would. And that's why you have visual hallucinations. One of the interesting aspects about this work is that what you're seeing with a visual hallucination, the primary elemental hallucinations of psychedelics, is you're actually seeing the primary processing of your visual cortex, which you've not ever seen except since when you were a child, a baby, but you don't remember that. So that's actually a rather elegant demonstration of how disrupting higher level processing can in a sense, show you the processing, which we know from other work, preclinical work in frogs and cats, etc. That's how the brain does reconstruct visual inputs. So that's a bit of the background science. Now, I just want to say a little bit about why we decided it would be interesting to study depression with psychedelics. And this is an example of translational medicine. When we started our work imaging psilocybin and LSD, we didn't expect to be treating depression. But two things came out of the imaging. The first was this. The first was that psilocybin reduced activity in this part of the brain, the front part of the brain, called the subgenual cingulate cortex. And that has been a long target for understanding depression. You can see in the green box a whole range of different treatments for depression were established by about 2010 as having an impact on activity in the subgenual cingulates. In fact, all of those treatments are associated with reduction in activity in that region. Even placebo, if you get better on placebo, there's less activity in that region, because that region is one of the key drivers for depressive thinking. And that's what led Helen Mayberg, the imaging data, led Helen Mayberg to then do deep brain stimulation in that region to switch it off and also lift depression. Well, psilocybin does that acutely, and so maybe it would lift depression by dampening down activity there. And then the other reason we moved into depression was because of the understanding that came out at about the same time of the role of this region called the default mode network in depression. So the default mode network is the network of the brain which does your thinking about yourself, about your present, your past, your future, your aspirations, your failures, etc. And it's called the default mode because it's only really active when you're not doing other things. So if you close your eyes and don't listen to anything and just reflect on your past or your future, then if we were imaging you during that, the default mode network would be predominant, as you see in the top image. But under psychedelics, the default mode is completely fragmented. It disappears. And that disappearance I showed you in a way with the MEG data. If you take away the activity in the posterior cingulate, the red blob at the middle at the back, then your sense of self disintegrates, and that's why people feel they float out the scanner and disappear into new dimensions, etc. So where you encode your sense of self is the default mode network, and that is disrupted by all psychedelics to our knowledge. What's that got to do with depression? Well, this remarkable study came out at about the same time as our psilocybin imaging study, showing that the default mode network is over-connected in depression. And not only is it over-connected, but also the magnitude of that over-connection is related to rumination, that process of negative thoughts which go round and round in people's heads they can't escape from. And the driver for that hyperconnectivity is actually the subgenual cingulate cortex, which is driving the hyperconnectivity and therefore driving the rumination as well. So that looked like a particularly interesting phenomenon. If we've got a drug which can disrupt the default mode network, and the default mode network is too active, maybe disrupting it in depression would lift depression. And this, I think, is exactly how it does work. So if we just go back to the image I showed you before, with the brain, how the brain and the visual system reconstructs what it expects to see. But in some cases, the brain, whether it's the visual system, not so much in depression, but other parts of the brain, the higher parts of the brain, in depression, create models of the world which are wrong, models of guilt, models of failure in depression. The cognitive processes of depression are obviously created in the brain, and they get out of control. So there's a miscreations of the brain. And I just want to use this quote from William Blake, who I imagine you'll find some of his pictures next door if you want afterwards. But Blake was a great visionary and philosopher, and he made the comment that man always sees with limitations, as if he's seeing the world through the chinks of a cavern. He doesn't see the bigger picture. And that is exactly how the brain works. The brain focuses on important things. It doesn't necessarily see the bigger picture. But in cases like depression and addiction, the brain creates a vision which is actually destructive and wrong. So where most of us might see at least a blue sky and white clouds, depressed people see a grey, miserable world, and addicts see their love objects. They see the bottle or the syringe or the white powder. And they get locked into that because their brain gets entrapped into a thinking process, which is often resisted by the person, but they can't stop. And there's another wonderful quote from Blake, which really sums it up, that humans make mind-forged manacles that determine their behavior. And I think psychedelics break these manacles and allow people to escape from those unwanted processes. And one of the aspects of the imaging work is highlighted here. So this is a bigger picture of those that increased network connectivity that I mentioned earlier on, under psilocybin and also under LSD, and we're about to publish the paper on DMT2. There's massively increased connectivity in the brain. On the left-hand side, you see what's called the small world brain. Your brain is very efficient because mostly it carries on doing what it's always done. Most of the connectivity is around the edge, the visual cortex talks to the visual cortex, auditory cortex to auditory cortex. Of course, there's got to be some cross-brain connectivity. If you see a bus about to run you over, you've got to move your legs to get out of the way. But mostly the brain saves energy by not doing a lot of this high-level, long-distance connectivity. Under psychedelics, the processes which constrain the brain to being very efficient and organized are broken down, and that allows this massive increase in connectivity. The connectivity, again, which your brain has never experienced since you were a child. And one facet of development and learning is to stop you having these wonderful imaginations in childhood and make you think in the same way as everyone else in society. The psychedelics put you back to where you were. You can think differently about things. We think our patients can get insights into the past, remember incidents which may have led to their depression. But more importantly, they can review their thinking and come up with new ways of thinking about their problems, about their life, and perhaps plan a new course so that they can come out of depression. And then on top of that, this neuroplasticity of psychedelics allows them not only to lay down those new thought processes can be laid down in a more, or consolidated in a way which means that they can then follow the new path rather than continually be stuck in the old rut of thinking. And I just want to finish by using this graphic to explain what we think is actually going on now. So this is from this recent paper of ours looking at global integration after psilocybin. On the left-hand side, in depression, the brain is very modular. The circuits of the brain, thought process, units of activity are very segregated from each other. It's rather like being in a very mountainous country, and you might be in one valley. Let's think about Switzerland, for instance. You might be in one valley in Switzerland, and you actually speak a language which is the different language to the next valley. And in depression, you get locked into a thinking process, and it's very hard to get out of because you're so deeply into that segregation. Now, under psychedelics, the landscape is completely flattened. That's what that huge entropy drive does. It flattens, it allows massive increase in connectivity. You can get out of your valley, and it stays somewhat flattened afterwards. And that is associated with, we think, the sense of increased cognitive ability and flexibility and control of people's thoughts that they have after they've had the treatment. And here's a couple of examples. And Ros Watts, who will talk about her therapy later on today, she's here. This is from her narrative paper from the first trial we did. 2 of our patients. My outlook has changed significantly. I'm more aware now it's pointless to get wrapped up in endless negativity. I feel as if I've seen a much clearer picture. Another patient said, my mind works differently. I ruminate much less, and my thoughts feel ordered, contextualized. Rumination was like thoughts out of context, out of time. Now my thoughts feel like they make sense with context and logical flow. And if you read that article, you'll see that lots of patient narratives which talk about how they've escaped from the routine of thinking which was destroying them. And just very briefly to show how we came to this conclusion of increased connectivity. So this paper was the first head-to-head of a psychedelic and an SSRI published last year. And it shows depression improving somewhat more with psilocybin than escitalopram. On the right-hand side, it shows well-being this sense of being in control of your life, much more improved with psilocybin than escitalopram. And I think perhaps it's the improvement in well-being which may, in the end, be more important for the long-term positive outcomes of psychedelics than necessarily the reduction in depression. Even though psilocybin did have powerful effects, particularly in terms of producing remission. You see the remission rates at the bottom there are much, much higher, at least twice, sometimes 3 times higher for psilocybin than escitalopram. But the well-being effect was very profound and enduring, and increased connectivity may well be a factor in that. So we now have I think we have quite a lot of evidence supporting the idea we developed a few years ago that there are 2 ways you can lift depression. You can lift depression with SSRIs. They work in the limbic system. They suppress the hyperactivity of the amygdala to stress, and they allow the limbic system to recover from the effects of stress. So SSRIs are to depression what a plaster of Paris is to a broken leg. You put the leg in plaster to allow the bones to heal. You put your brain in a plaster with an SSRI to allow your limbic system to heal from the chronic stress that leads to depression. But the downside of that is that protection that SSRIs give to the limbic system also comes at a cost. It comes at a cost of not being able to be as reactive to positive mood. So you get a kind of a stabilization of mood from both ends. And some patients don't like that. They often complain of having blunted mood. The activity of SSRIs is through the serotonin 1A receptor, the major inhibitory serotonin receptor in the brain, which can dampen down that limbic activity. Psychedelics, we know, work through the 2A receptor, and that's in the cortex, as I've shown you. They disrupt cortical activity. They fragment the tramline thinking, the ruminations of depression, and they increase the cortical connectivity and cortical flexibility, and that's what leads to the lifting of mood and also this improvement in the sense of well-being, more connected with the world, more connected with other people. It breaks that hyper connection with the inner thinking, the ruminations. And I'll just finish with this quote from George Bernard Shaw, a great playwright and philosopher. Those who cannot change their minds cannot change anything. And what I think we have shown, and others of course as well, is that treatment with psychedelics has changed the minds of our patients. And I think it's done that through changing their brains. And I just hope that this research, and all of you here, I'm sure, will agree with me in that we want to change the public's attitude, and we want to particularly change politicians' minds so that we can actually bring these remarkable therapies to the people that need them. Thank you.
David, thank you.
We'll take questions now.